Good day, everyone. You're welcome to my YouTube channel, Everything Medical. On today's class, I'll be treating transport of fatty acid, which is the carnitine shuttle, under metabolism. Before we delve into the transport of the fatty acid, first of all, let's give a background study on carnitine. So, what is carnitine? What is carnitine? This carnitine is a very essential transporter of fatty acid. Carnitine is synthesized into organ. That means the organ of synthesis or biosynthesis of carnitine, it involves two major organs, which is the liver and kidney. Carnitine is synthesized in the liver and the kidney from two major amino acids, which is the lysine and methionine. Two major amino acids, which are lysine and methionine. Carnitine is synthesized from lysine and methionine in the liver and the kidney. What is the essence of this carnitine? The carnitine tends to transport the activated fatty acid, which is the acetyl-CoA, from the cytosol to the mitochondria. Why? Because beta oxidation of fatty acid occurs in the mitochondria, and the major metabolite that features in beta oxidation is that activated fatty acid, but then it is not permeable to the mitochondrial membrane, so it requires a transporter called the carnitine. Now, we have stated the organ synthesis of carnitine and the two amino acids that tends to produce carnitine. Now, let's give the chemical structure of carnitine. Chemical structure is the chemical structure of carnitine. It goes this way. This is the chemical structure of carnitine. Now, let's go to the chemical name of carnitine. Chemical name of carnitine. It is chemically called beta hydroxyl gamma trimethyl ammonium butyrate. So this is the chemical name of carnitine. Why this is the chemical structure of carnitine? These two are the two amino acids in which carnitine is being synthesized from. Why the organ of synthesis of carnitine is the liver and the kidney. During pregnancy or during growth, the requirement of carnitine is always high in production. But then, when there is human genetic mutation whereby the carnitine is being deficient, there is always impairment in fatty acid oxidation, because there is no transporter that is available to transport the activated fatty acid to be channeled to beta oxidation so that it will produce acetyl CoA, which goes to the TCA cycle. Automatically, by saying so, it is automatically meaning that carnitine aids in combustion of fatty acid, or the burning of fatty acid to give energy or to produce energy. To tell you that carnitine is very, very essential in fatty acid metabolism or in fatty acid combustion to produce energy. Then let's dive into how the carnitine transports the activated fatty acid from the cytosol to the mitochondria. As you can see on my diagram on the board, I have the cytosol. Here are the cytosol, while here are the mitochondria. Look at the mitochondrial membrane. Now let's go. We have the carnitine. Here we have acyl CoA. Acyl CoA. Here we have the CoAs. This acyl CoA is called the active fatty acid or the activated fatty acid. Recall that fatty acid activation occurs in the cell cytosol. I've done it in my previous video. You can go and watch it and get the detailed explanation on how fatty acid is being activated in the cytosol. So as you can see here, the enzyme responsible for step 1 is carnitine acyltransferase, or carnitine palmitoyltransferase. So this enzyme can be called carnitine acyltransferase or carnitine palmitoyltransferase. So we can call it CAT1. CAT1, which is for carnitine acyltransferase in a short form. Now, what is the function of this enzyme? This enzyme, what does it do? It transports the hydroxyl group of carnitine to the acyl group of acyl CoA to form acyl carnitine, to form acyl carnitine. To form acyl carnitine. That is the work. It helps to transfer the hydroxyl group of carnitine to the acyl group of acyl CoA to form acyl carnitine. This enzyme is called in the short form CAT1, which is carnitine acyltransferase, or it is alternatively called carnitine palmitoyltransferase. So once this enzyme has formed acyl carnitine by the combination of the functional group of carnitine and acyl CoA, another membrane carnitine exchange transporter called translocase will transport it from the cytosol to the mitochondria. That enzyme is called translocase enzyme. Translocase enzyme. It is alternatively called carnitine acyl carnitine translocase. What is the work? It transports the acyl carnitine from the cytoplasm or the cytosol to the mitochondria. Here we have our acyl carnitine. So this enzyme is called a membrane carnitine exchange transporter. It transports the acyl carnitine from the cytoplasm or cytosol to the mitochondria, as you can see it here. So once it has transported it to the mitochondrial part of the cell, another enzyme known as CAT2, which is carnitine acyltransferase 2, will transport it, or it will help to cleave it so that it will give a certain product. Let's dive into. So here we have another enzyme called carnitine acyltransferase 2, which is called CAT2. It is called CAT2. It can be alternatively called carnitine palmitoyltransferase as well, as we said in this CAT1, called carnitine palmitoyltransferase. What is the work of this enzyme? This enzyme cleave the functional groups from this acylcarnitine. That is the work. Now we have the CoAs. Finally, we have our activated fatty acid back. As you can see it, acyl-CoA. This is our activated fatty acid. As you can see, it was in the cytosol, now it is now in the mitochondria. Without the help of the carnitine shuttle, it wouldn't have been transported so that it will feature in beta oxidation. So from here, to form acetyl-CoA, beta oxidation has to take place. We have our beta oxidation. So for acyl-CoA to form acetyl-CoA, it must undergo beta oxidation, because the essence of beta oxidation is to produce acetyl-CoA, which enters the TCA cycle to be oxidatively decarboxylized as a 2 atom of CO2 in step 3 and step 4 of the TCA cycle. That means acetyl-CoA here goes down to form the TCA cycle. So that is the work. So once this enzyme has cleaved acylcarnitine to form CoAs and acyl-CoA, it will as well form carnitine, because what that form acylcarnitine is the hydroxyl group of carnitine and the acyl group of acyl-CoA. So here we have our carnitine. Now, once carnitine has been formed in this place, a membrane carnitine exchange transporter known as translocase or carnitine acylcarnitine translocase will transport the carnitine from the mitochondria back to the cytosol. So it goes in a cycle. If carnitine should be deficient, automatically there will be no beta oxidation. And once there's no beta oxidation, there will be production of what? Acetyl-CoA. That means the person will always be in a hypoglycemic state, because the energy that is needed for tissue respiration will be channeled in glucose pathways, because fatty acid energy helps as well in tissue respiration. So that is how it works. Now let's take the difference between acyl-CoA and acetyl-CoA. Acyl-CoA and acetyl-CoA. What is the difference between acyl-CoA and acetyl-CoA? Acyl-CoA is a combination of acyl group of any fatty acid ranging from carbon number 4 to 26, ranging from carbon number 4 to carbon 26 in combination with coenzyme A or CoA. While acetyl-CoA, which is they follow the TCA cycle or the chief metabolite, is a combination of acetate or acetic acid with coenzyme A. So that is the difference. This is the chief metabolite, a metabolic biochemistry, while this is the activated fatty acid which will undergo beta oxidation to produce acetyl-CoA, which is this. So this class, I've stated the differences between acetyl-CoA and acetyl-CoA. Watch out for my next class on beta oxidation of even chain fatty acid. Thanks for watching.